Good day, this is the Professor, CBMD PhD, and today's Tuesday, September 12th, 2023. It is 4:59 and 9 seconds, AM Eastern Standard Time, and today's research, cell therapy. So let's jump to it here. The CAR T-cell therapy, without side effects, researchers show, results in preclinical models, date September 7th, 2023. Source: Medical University of South Carolina. As some patients have experienced incredible recoveries after CAR T cell therapy, but they've also endured incredible strong and scary side effects, in part because of the lymphodepleting chemotherapy that is performed before the CAR T cells are reinfused. What if CAR T cell therapy could be performed without lymphodepleting chemotherapy? As CAR T cell therapy is currently used to treat some types of blood cancers that have returned after treatment or haven't responded to chemotherapy, it is both expensive and extensive. Some of a patient's T cells are removed and sent to a laboratory where in the process that can take several weeks. They are engineered to add chimeric antigen receptors, CAR, that are tuned to home in on specific proteins on the surface of cancer cells. The newly formed CAR T cells are then reinfused into the patient to attack the cancer. As some patients have experienced incredible recoveries after CAR T cell therapy, but they've also endured incredible strong and scary side effects, in part because of the lymphodepleting chemotherapy that is performed before the CAR T cells are reinfused. Lymphodepleting chemotherapy kills off existing T cells to create a blank slate for the CAR T cells, and research has shown that the therapy is more effective after lymphodepleting chemotherapy. They show that encoding CAR T cells with instructions to create a hyperactive form of protein state 5 prompted the CAR T cells to engraft or take root and begin multiplying without requiring lymphodepletion. In their experiments, the engraftment process was cell autonomous, meaning it didn't depend on the surrounding environment, but happened solely because of the instructions from within the cells. They present a lot of evidence to support that notion that it is a completely cell-autonomous process, and that it is fundamentally driven by activation of STAT5. And so by transiently activating STAT5 during that phase of adoptive transfer, the initial engraftment phase, you are tricking the cells into essentially thinking they are going into a lymphodepleted environment. And so they engraft and become very functional and do everything they are supposed to do. Once they saw that they didn't engraft, they just benchmarked how they function with as many different benchmarking assays as they could, comparing it to conventional adaptive transfer using lymphodepletion as their benchmark. The scientific investigators and microbiologists said they came to STAT5 because of its role in the cytokine signaling pathway. It has been known for some time that the interleukin cytokines IL-2, IL-7, and IL-15 are instrumental to the engulfment process, and some have suggested injecting patients with IL-2 or IL-15 in place of lymphodepleting chemotherapy, but each of those cytokine pathways requires STAT5. They reasoned that they could recapitulate an entire signaling process at the node of STAT5 rather than trying to tickle it up at IL-15, IL-2 receptors. And by doing that, they also have more of a cell-autonomous effect where they don't have to expose the patient to a bunch of IL-7, IL-15, and IL-2, which can be dangerous. The team used messenger RNA transfection to implant the instructions within the CAR T cells for activating STAT5. In preclinical models, they found that their method reduced cytokine release syndrome, sometimes called cytokine storm, one of the most serious side effects. The CAR T cells carrying super STAT5 also got cancer under control and appeared to create memory cells trained on that cancer. The scientific investigators and microbiologists said that's eliminating the need for lymphodepleting chemotherapy to mean that CAR T cell therapy would become viable for more types of diseases. The balance of harms and benefits The lymphodepleting chemotherapy is different for someone facing a recurrent lymphoma that no longer responds to chemotherapy than for someone with chronic condition like lupus. You might be able to treat some of these more serious lupus cases. Doctors would never want to lymphodeplete somebody with lupus with the combined chemotherapies, fludarabine and cyclophosphamide, but if we don't have to lymphodeplete them anymore, then you can imagine trying out this therapy with them. Eliminating lymphodepletion can also change dosing schedules. Now you can imagine re-administering dose after dose to the patient instead of one and done. If you don't have to lymphodeplete them, you might be able to just give them an injection every month. The scientific investigators and microbiologists are working with the MUSC lupus erythematosus, a research group of potentially developing clinical trials. They are also talking to biotechnology companies that work in CAR T-cell realm and have the ability to launch new clinical trials. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.